Welcome to CEC-UGC lectures. I am Dr. Kumar Shantanu from Department of Botany, Deshbandhu College. We have started our discussion with various aspects of biology of pteridophytes. In this lecture series on archegoniates, we have already covered various genera, various life forms, ecological aspects and economic importance of the first land plant, that is pteridophytes. We are continuing our discussion with the first vascular land plant group. We also call them pteridophytes. As we have seen in our previous lecture, that pteridophytes they have a very distinct structure called as vasculature, made up of xylem and phloem tissues. For the first time, heterospory has set in different genera of pteridophytes. We have also seen an overall brief life cycle of a pteridophyte. In this lecture, we shall continue our discussion with the formation of spores. So, most of the pteridophytes are homosporous. That means they produce only one type of spores. However, few species are also found which are heterosporous. That means they produce two types of spores, different in their size. The smaller size spores are called as microspores, whereas the larger sized spores are called as megaspores. In heterosporous species, the arrangement of microspores and megaspores may vary. Sometimes the arrangement of microspore is found in one entire strobilus, whereas the other strobilus contains only megaspore. In some of the cases, we also find presence of microspore and megaspore within the same strobilus. They could be separated laterally or they could be separated from apical and posterior positions. These spores on germination produce gametophyte. Amongst heterosporous species, the microspore give rise to microgametophytes, whereas the megaspore gives rise to megagametophyte. These microgametophyte and megagametophyte, they ultimately give rise to gametophytic thallus. The smaller gamete or the smaller gametophyte gives rise to male sex organ, that is antheridia, whereas in the case of megagametophyte we will find the presence of female sex organ, that is archegonia. The gametophytes of these heterosporous forms are retained within the spore coats. That is, they are endosporic, whereas in homosporous the gametophytes are exosporic. That is, they are formed outside the spore walls. Now, for the first time during the evolution of plants, we could find a distinction between homosporic and heterosporic development of gametophyte. Let us try to understand what is a what is a exosporic gametophyte and what is an endosporic gametophyte. This diagram shows a distinction between two different types of gametophyte. The gametophyte in Lycopodium shows exosporic type of gametophyte, whereas gametophyte in Selaginella shows endosporic gametophyte. Now, in case of exosporic gametophyte, the spore starts germinating, the spore divides and gets converted into multicellular structure. The spore wall gradually opens up. The spore wall gradually opens up and the gametophyte comes out of the spore wall, and in this gametophyte male and female sex organs are born. And here we can see a developed gametophyte of a club moss, that is Lycopodium. Whereas in case of endosporic gametophyte, the spore divides mitotically, gives rise to multicellular gametophyte. These gametophyte are formed inside the spore walls. Inside the spore wall only, the male gametophyte will give rise to antherizoid. Here we can see multiflagellated or biflagellated sperm cells are produced, whereas archegonia are formed on the multicellular gametophyte, which is found within or inside the spore wall only. And the process of fertilization takes place inside the spore walls. So here in case of gametophyte of a spike moss, that is Selaginella, we could see an endosporic gametophyte, whereas in case of club moss we could see exosporic gametophyte. In general, pteridophytes form green, dorsiventrally differentiated thallose gametophytes with sex organs restricted on the ventral surface. The sex organs may be embedded or they could be projecting. The female reproductive structure is an archegonium and the male reproductive structure is an antheridium. In a typical gametophyte of pteridophytic genera, the female sex organ, that is archegonia, are found located towards the growing point, that is your apical notch. Whereas antheridium, that is male sex organ, they are found towards the posterior margin, towards the posterior end of the gametophyte. From the posterior margin, rhizoids are also developing, which provides anchorage to the entire gametophyte and also sometimes help in absorption of water. The archegonium has invariably 4 longitudinal rows of neck cells whose height varies in different genera. The antheridium, whereas, consists of a single layer of sterile jacket of cells enclosing a mass of androcytes. These androcytes are also called as antherizoid mother cells. The opening of the mature sex organs and the subsequent fertilization is still conditioned by the presence of water. Here we could see the similarity with bryophytes. In bryophytes also we have discussed that the fertilization is mediated by the presence of water. In case of pteridophytes, the archegonium is having short neck, 4 longitudinal rows of cells give rise to neck. Inside the venter, in the center, is found the egg. The antheridium is formed of a single layer jacketed cell, inside which are found sperm cells. These sperm cells are flagellated. In some genera, the sperms are multiflagellated, whereas in some genera, some primitive genera, they are still biflagellated. So, the movement of sperm towards the archegonia and finally up to egg cell is mediated by the presence of water. Here we could see the structure of an archegonia and antheridium in much detail. You could very well see the jacket layer is made up of a single cell layer. Inside the single cell layer jacket develops multiflagellated numerous sperm cells, whereas the number of egg cell per archegonia remains one only. The sporangia can be distinguished in two types, a eusporangium and a leptosporangium. This distinction is based on their developmental stage. In case of eusporangium, the sporangium originates from a group of initial cells. Such type of sporangium are found in Xylotum and Selaginella, whereas in case of leptosporangia or leptosporangium, their origin is from a single initial cell, and such type of sporangia are found in almost all ferns. Let us understand the distinction between leptosporangiate and eusporangiate sporangium with the help of diagrams. In case of eusporangia, one of the initial cells of sporophyll divide, and it divides and multiple cells of the sporophyll take up the role of sporangia primordia. These cells, these sporogenous cells, give rise to a sporogonium with tapetum. These sporogonium with tapetum contains sporocytes, and these sporocytes finally give rise to sporangium. So, in this condition, the development of sporangium from the sporophyll cells takes place, and multiple cells of the sporophyll are involved in the development of sporangium. On the contrary, when we talk about leptosporangium, a single particular cell on the epidermis of sporophyll acquires the role of sporangium primordia. It divides and gives rise to an outer cell. This outer cell on the surface of sporophyll then further multiplies in various planes, giving rise to a sporangium which has an outer jacket layer, and inside this jacket layer resides multiple sporogenous mother cells. These spore mother cells will give rise to the spores proper. So, in this case of leptosporangia development of sporogenous sporangia, we see that a single cell of sporophyll is involved in the development of sporangia. So, eusporangia differs from leptosporangium in the number of cells of sporophyll involved in the development of sporangium. So, if we could sum up the life cycle of a fern, we could observe that a gametophyte bears archegonia and antheridia. From the antheridia arises numerous multiflagellated sperms. These sperms could be coiled, or the number of flagella could be 2 or more than 2. After fertilization, zygote is developed. Zygote multiplies mitotically to give rise to embryo. These embryo are the representative of sporophytic generation. They further develop to give rise to aerial sporophyte. These sporophytes have distinct root, stem and leaf. On the leaf, special structures are born called as sporangium. These sporangium are born in clusters. These clusters of sporangium are called as sori, and the leaf which bears this sori, they are also sometimes termed as fronds. From these sporangium, spores are released after their dehiscence. These spores are formed after meiotic division of spore mother cells. So, these spores further germinate and give rise to a haploidic gametophytic generation of pteridophyte. So, thus this entire life cycle is complete, where sporophytic generation alternates with the gametophytic generation. The gametophytic generation is short-lived and very small in size, whereas sporophytic generation is very long-lived and nutritionally independent and dominant phase of the life cycle of pteridophyte. Now, let us talk about the classification of pteridophytes. As far as the classification of pteridophytes is concerned, we could classify pteridophytes on the basis of morphology, structure and details of reproductive structures. They can be divided into 4 distinct groups. They could be named as Xylophyta, Lycophyta, Anthrophyta and Filicophyta. Now, some of the plant biologists consider these groups as class, and some also consider them as division. So, in division Xylophyta, there are 2 class which could be distinctly found. One is called as Xylopsida and the other one is called as Xylotum and Rhinia. As far as division Xylophyta is concerned, this division has both living as well as extinct plant groups. Vascular system is of very similar type and it is divided into 2 classes. The first class is called as Xylophytopsida. Now, Xylophytopsida has all the extinct members which are known as by the name of Eglophyton, Horneophyton, Cooksonia, etc. Only a sporophyte is known of this class. The main characteristic feature of this class are the plant body is divided into underground rhizome and aerial naked branches. Now, in this class, proper root is still not discovered because all the plants which are extinct, they have been clubbed under this class. In place of roots, the unicellular rhizoids are found in tufts on the rhizome. In this class, the sporangia are terminal or lateral in position. They are all homosporous, and in the vascular system, the central solid core of xylem tissue is surrounded by phloem. The second class is Xylopsida. This class is represented by 2 genera, that is Xylotum and Mesipteris. The main characteristic feature of this class happens to be the sporophyte is divided into 2 parts, the underground rhizome and the aerial dichotomous branches. Rhizomes are mycorrhizal. Rhizomes have mycorrhizal association of fungal hyphae. The aerial branch contains some leaf-like appendages. Stele is protostele. We will talk about stele in much greater detail in our coming lectures. The sporangium is homosporous. The enterozoites are multiflagellated, and similarly, the second division of pteridophytes is termed as Lycophyta. Now, this division has both living as well as fossil members. It includes 5 living members, that is Lycopodium, Phyloglossum, Selaginella, Stylites and Isoids. The main characteristics of Lycophyta is that sporophytic plant consists of roots, stems and small microphyllous leaves. The plants could be homosporous or it could be heterosporous also. The enterozoites are biflagellate or multiflagellate. In case of division Sphenophyta, it is represented by only one genus, that is Equisetum. In case of Sphenophyta, the sporophyte is differentiated into roots, stem and leaves. The stem is jointed, solid and protostelic. Some members do show secondary growths also. Sporangia are found in terminal cone strobilus. Enterozoites are multiflagellate. Spores contain elaters. The anatomy of the stem of Equisetum shows a combination of hydrophytic and xerophytic characters. The fourth division of pteridophytes, the third division, that is Sphenophyte, they also have nodes and internodes which are differentiated in the internal structure. In transverse section, internode differentiates into epidermis, cortex, endodermis, pericycle and vascular cylinder. In Sphenophyta, epidermis is encrusted with heavy deposition of silica. The cortex is differentiated into outer sclerenchymatous and chlorenchymatous and inner parenchymatous region having large schizogenous canals called as vallecular canals. Cortex is followed by single layered endodermis. The fourth division of Pterophyta the fourth division is Pterophyta. The prominent features of Pterophyta are the sporophytes are differentiated into rhizome, roots and leaves. The rhizome could be creeping or it could be erect with aerial stem. Leaves are mostly dimorphic. Sporangia are found in the sori. Stele may be protostelic, siphonostellic or dictyostelic. Some members are Ophioglossum, Helminthos, Tachys, Nephrolepis, etc. They bear vessels in the xylem also. So, till now we have covered the classification of pteridophytes. We shall try and understand the origin and evolutionary aspect of pteridophytes and various different groups in our next coming lecture. Thank you so much.